to have Nikola Stikov join us today to actually give his talk. He's an associate professor of biomedical engineering, and and a researcher at the Montreal Heart Institute, and a co-director of Neuropoly, which is the neuroimaging research lab at École Polytechnique and University of Montreal. His research runs the gamut of quantitative MRI, from basic issues of standardization and accuracy to biophysical modeling, microstructural imaging in clinical application. And what he's going to talk to us about today is thinking outside the black box, a vendor neutral approach to quantitative MRI. So go ahead, man.
Perfect. So thank you for the invitation, Borjan. It's really good to be here virtually. And it's exciting to tell you about a project that's still in its early stages, but I think it would be excellent to get your feedback on a lot of the issues that we're trying to tackle. And let me just share my screen. All right, I hope you can all see my slides. Perfect.
Yep, it's all good.
All right, so I decided to first tell you a little bit about my career path. I did my undergrad, master's, and PhD at this lab at Stanford, the Magnetic Resonance System Research Lab. I imagine a lot of you recognize some familiar faces. Here's Al Makovsky, the founder of the lab. And this is my family attending his 90th birthday celebration. Mickey Lustig right there, probably a lot of you know him. Dwight Nishimura, John Pauly, my co-advisors. And I would like to just give credit to this lab for teaching me one very important equation. This is the signal-to-noise ratio equation for MRI, but I often use it as a metaphor for taking your time. Here are people that have spent a long time in this lab, not really publishing frequently, but when they do, they publish high-quality work. And I think it's a testament to the way that Al ran this lab, that we were not afraid to take twice the time necessary or 4 times the time necessary to get the signal to noise twice as high. I think I benefited from that philosophy because I took my time with research. This is my timeline. I tend to subscribe to this slow science movement where we don't publish too often and try to think about how we can present our work so that it's not really salami sliced. There is an interesting blog post that just came out a couple of days ago by Yoshua Bengio, one of the big AI people that's currently at the University of Montreal, promoting this approach. And I really hope that at least some scientists follow this kind of slow, methodical grind through science. This is the first paper that I ever published. It's in 2011, and it's actually 2 years after I completed my PhD at Stanford. I have a lot of abstracts from the past, but I never thought that it was necessary to get a 10-page PDF out. But then this came out in 2011, kind of steered the rest of my career. It was about measuring the myelin-G ratio. In 2015, we followed up with an experiment that was pretty well received. Then we shared the data, we shared the code that came together with that work. And finally, we wrote a review article about 7, maybe 8 years after the original paper appeared. And partly I'm showing you this because it's important to emphasize how G ratio is the bulk of my work until now, but also to tell you that I will not be talking about G ratio today. And if you ask me, even these 4 papers, I'd make them be 1 paper. I would say, this is the pre-registration, this here is the experiment, this is the data and the code, and this is kind of the story that we tell afterwards. But I think that might be pushing it too hard, and I probably would never have gotten a faculty position if I did that. So anyway, there it is, the 4 papers that kind of got me to a point where I'm now tenured, I'm at the University of Montreal, I'm currently on sabbatical in my home country, Macedonia. So I guess things have worked out. And I can't say that it's an easy path, but at least what you're gonna see for the rest of the presentation is hopefully me practicing what I preach. Um, so because I take my time in also building community. I have been involved with the Journal of Magnetic Resonance in Medicine for a long time. I was on its board for about 4 years, recently stepped down. I was guest editor of a special issue for Neuroimage on imaging brain microstructure. I started the blog of the Organization for Human Brain Mapping, also the blog Magnetic Resonance in Medicine Highlights. This is actually Al Makovsky on the cover of one of the magazines. And this is something that I recently passed on to a software developer in my lab. The blog of the ISMRM, this organization that I'm very, very proud of, and particularly because Borjan is with us today, we started this together in 2008. It was an effort to bring magnetic resonance research in the Balkans. Still haven't given up on that idea. These seminars happen regularly. We've had one in Turkey, one in Slovenia, one in Bulgaria, one in Turkey, I said that. And Lana, we should do one in Croatia after the pandemic. And then I'm going to tell you a bit about the Canadian Open Neuroscience Platform, which is this pan-Canadian $11 million grant where I'm on the steering committee and I'm also in charge of its publishing platform. So because I'm talking about quantitative MRI, I'm not sure whether all of you are familiar with the topics. So I just have this very brief slide that talks about the difference between conventional and quantitative MRI. And when I talk about conventional MRI, we talk about an alphabet soup of contrast mechanisms. And there we are, we take a spoonful, we get some T1, we get some T2, and out comes an image that's weighted. In this case, it's a T1 weighted image, but a lot of these other contrast mechanisms also contribute. When we talk about quantitative MRI, well, then we talk about compartmentalization, different kinds of contrast mechanisms, all in their nice compartments in like a Japanese style bento box. So here it is, the T1 compartment, and let's see what it would look like. Well, it looks like something we call a T1 map. T1 weighted image, T1 map. The contrast is inversed, but I don't think that makes much of a difference. The images are digital, except the numbers here don't make much sense. They could be all over the place. It's up to us to play with the dynamic range to get some kind of contrast. Whereas here, in this case, in the bento box, the numbers matter. These maps have units, and when we hover over a point, we can find out what is the exact number for the T1 value in white matter, gray matter, CSF, and then hopefully we can repeat these numbers across different vendors, compare them between subjects, or follow a single subject longitudinally to see how the numbers have changed. So it's a nice story. That's the reason that I got excited by quantitative MRI. I really think it adds something to the existing paradigm. But the more I was doing my research, the more I realized that we have problems with T1 mapping. And I realized that kind of late in my PhD. This is a paper that was published by Joëlle Barral, where she wanted to come up with a robust methodology for in vivo T1 mapping. Something that has been around for a long time, but never really properly formalized with the code and the necessary methodology to go along with it. This paper came out in 2010, and then it was the basis for another paper that I published in 2015, which was called On the Accuracy of T1 Mapping, Searching for Common Ground. Now, this paper is the only work that I've ever gotten rejected by the ISMRM. It was not accepted as an abstract, so I actually had to write up the paper. And eventually, it actually became one of the top 5 most cited papers in the journal. And I think when I show you the results, you'll realize why. Basically, what I did is I used Joëlle's approach for a robust inversion recovery T1 mapping method. And then I said, well, let's take a look at how the T1 parameter varies at 3 Tesla in white matter in 7 different studies. And here you have the inversion recovery gold standard. And here you have something called look locker T1 mapping. And here you have something called variable flip angle T1 mapping. And I don't want you to stare at this too long, but just realize that the T1 values vary from 690 all the way to about 1100, which is a variation of about 70%. And that is not a realistic variation. This is all method dependent. The T1 parameter at 3 Tesla in white matter is actually a very narrow peak. So we said, let's repeat this, but now let's do it at the same site in the same session. Inversion recovery, look locker, variable flip angle. Let's see what happens. There's a T1 map for inversion recovery. Here it is for look locker. Here it is for variable flip angle. These are just 3 different ways of measuring the T1 parameter. And now let's take a look at the histograms. Well, there's variability. Here you go, the gold standard makes the T1 value be around 800, 830 milliseconds. Variable flip angle, slightly higher peaks. Look locker, slightly lower peaks. This is maybe, expected, but at least people were surprised to see it. And now this paper is kind of making people aware we have a problem with T1 mapping. Let's kind of expand more and see what we can do about it. Here's a beautiful work by Eugene Lee and Zoltan Nagy in Zurich that talks about the intra and inter-vendor reproducibility of T1 at 3 Tesla. And I'm not going to show graphs, I'm just going to read a sentence from it. But basically, they observed a consistent bias in their comparison in that the T1 relaxation time estimates were 8 to 10% higher on average when calculated from data acquired in the Philips scanner compared to that from the Siemens scanner. Here's another article, very recent, less than a year ago. And this is How Stable is Quantitative MRI? Assessment of intra and inter-scanner model reproducibility using identical acquisition sequences and data analysis programs. And that's the key, identical. If they're identical and they're on the same scanner, then the variations are very low. Inter-scanner variations are on the order of about 5%, but then the acquisition sequences need to be the same and the analysis needs to be the same. And that provided that identical acquisition sequences are used, discrepancies between qMRI data acquired with different scanner models are low. So, maybe a little bit more optimistic. Here's another one. This one is actually coming from the NIST, the National Institute for Standards and Technology. And this is work by my colleague, Catherine Keenan, with whom we actually did our PhDs together. So we worked on cartilage imaging before we moved on to quantitative MRI. And this is also an important observation. When scanners get upgraded, which is very often, the T1 values vary. And usually the contributions come from B1, and the variations are on the order of 5% to 30%. So we have a problem, especially because scanners don't often tell you what they do after the upgrade. So it's kind of like a black box. What we tried to do is get people to use this original paper by Joel Barral, Inversion Recovery, Gold Standard T1 Mapping, and perform T1 mapping on a NIST phantom, which is a standardized phantom that's identical across sites, and then also in humans. This is a challenge that's organized by a software developer in my lab, Mathieu Boudreau. Here you can read a little bit more about the challenge. We got quite a lot of submissions. We got 19 submissions, of which many of them were multiple acquisitions. So we had 41 data sets of the NIST phantom, which is the standard phantom that you'll also see in a little bit. 56 of them were on human subjects, and most of them were at 3 Tesla, except for one phantom, which was at 350 millitesla. And then all 3 vendors were represented, GE, Philips, and Siemens. We created a beautiful dashboard that was built by my student, Aga Karakuzu. On this dashboard, you'll see that we have the phantom and the brain data, and you will see all of the centers that actually contributed to the challenge. And you'll see what fraction were Philips, Siemens, and GE, which fraction were phantoms, which fractions were brain. And this is what this dashboard looks like. It's actually a beautiful app, and it guides you through different parts in the phantom and the brain where you can take a look at the stability of the results. Well, the results were very similar to what we have observed in the past. And here it is. In brain, the variations tend to be a little bit higher than in phantoms, which is, again, something that we expect. But finally, we have a large collection of data that people can analyze and try to decide what are the factors that are actually affecting these variations in the T1. Given that the presentation is short, I'm not going to dwell on these results except to tell you the link is there if you want to explore it. And if we have some time or if you have questions, I can guide you through the dashboard and show you some of our findings. Now, all of this is brain, and brain is complicated enough as it is. Cardiac T1 mapping is even worse. We recently wrote a book chapter, again, with the same researcher, Joel Barral. She's currently at Google, but she still likes to do MRI work. So we wrote a book chapter for an edition by Springer, edited by Phil Yang. We also have a paper talking about the differences in different cardiac magnetic resonance sequences for native myocardial T1 at 3 Tesla. And the results are good in phantoms, not so good in humans. And you'll see that, again, 3 methods, one of them severely overestimating the values compared to the other 2. And I thought, well, maybe this is a good chance to write the same kind of paper that I wrote for brain T1 mapping. Let's just do it in cardiac. And I called it, let's talk about cardiac T1 mapping. Many of the usual suspects from these previous two papers were co-authors. We thought it was excellent work. We thought it was going to get the field straight. But we got the worst reviews I've ever read in my entire career. And basically, one of the authors, one of the reviewers called the paper irritating. And the other reviewer, on a scale from 1 to 10, 1 being the worst paper ever, 5 being the best paper ever, gave us 1s in every single category. So this is never going to turn into a paper. I'm okay with it. It's going to be a preprint on bioRxiv. And hopefully at some point, cardiac MRI folks realize that they have the same problem that brain T1 mapping folks have. Unfortunately, for cardiac T1 mapping, these sequences are actually products on the scanners. So they're black boxes, people use them. Some people even use them for diagnosis, and that's scary. Why is it scary? Because quantitative MRI is pointless if we cannot peek inside the black box that generates the numbers. It's different for qualitative MRI. You see a weighted image, you see some contrast, you can determine what kind of a tissue you're talking about. You have numbers, unless it's clear how the numbers come, and if the numbers don't reproduce, we have a big, big problem. So I was recently elected to lead the ISMRM Reproducible Research Study Group, and we did a survey. This survey was, broadly speaking, from your experience, do you feel there is a reproducibility problem within your area of expertise? Now you might say this is a biased sample. These are people that care about reproducibility. We tried to advertise broader, but you never know who got reached. We had 70 responses, so again, not very representative, but about 3/4 of the people said, yes, we do, and then another 20% said maybe. And then when we were asking, what are the common problems that you encounter related to reproducibility? Many people said general lack of detail, no code available, systems too different between sites, and the data not available. These were the top 4 concerns that people had about reproducibility. And again, if you want to see more, here's the link. You can click on it, and it will take you to a summary of the questionnaire and some analysis. So we decided to do an analysis of articles published in MRM, in magnetic resonance in medicine. And again, this was done by Mathieu Boudreau in my lab. And what he did is looked at all the papers published in 2019, actually January to November 2019, which were about 470. And then he did a keyword search for all of these aspects of reproducibility and open science that could help identify papers. He identified 174 that matched at least one keyword. And then out of those, only about 52 actually linked to share code and data. So that's about 11% of total papers. The most common coding language was MATLAB. And then the most common location where the code is hosted was GitHub. And then only 12% of the papers actually shared enough to be able to reproduce a figure from the paper. Right around the time when this analysis was happening, I was also writing an editorial for MRM. I wrote it with Josh Trashko, who was a fellow member on the board, and Matt Bernstein, who was the outgoing editor-in-chief for the journal. And the name of the editorial is Reproducibility and the Future of MRI Research. And I will just read one quote from that article. For a truly international journal like MRM, it will be difficult to find a one-size-fits-all answer. However, few would disagree that reproducibility accelerates scientific progress, as well as its translation to clinical practice. As tectonic technological changes shape research in the 21st century, we need to be careful not to damage its foundations, and instead focus on aspects of open science where we can reach consensus. The open science movement needs to pick its battles. The battle for reproducible research is one that it cannot afford to lose. So when I talk about open science, I talk about reproducibility. The other aspects of open science are really, really important, but my battle, the battle I've picked, is reproducibility. What is our contribution in this battle? Well, we've built a piece of software. It's called qMRI Lab. And the idea is to bring all of quantitative MRI under one umbrella. It is open source, and it has quite a lot of contributors. These are the key contributors, and the lead author is my student Aga Karakuzu, but many of these folks are past members of the lab. Julien Cohen-Haddad is my co-PI in the lab. Ilana Lippert is a colleague from McGill, and Bruce Pike is my former postdoc supervisor currently at UC Calgary. And here's the article that just came out. It's published in the Journal of Open Source Software. So it's not a PDF article. It really is a code article. The review process was beautiful. Everything is transparent. We were very, very happy with the way they treated us. And here's what's under the hood of qMR Lab. We gather available tools. We collect code that has been peer-reviewed in other articles. The code base is modular. It's published under an MIT license, so anybody can use it. And it's collaborative. At the moment, we have about 20 contributors. We have simulations, we have data fitting, we have noise and sensitivity analysis, and we also have a graphical user interface, and we can perform batching. Which methods? Well, the bread and butter: diffusion, MRI, relaxometry, magnetization transfer, B1, B0 mapping. And finally, to deploy it, we have a version in Octave, which is open source. We have a version in MATLAB as well, which is compiled. We use different kinds of containerization platforms to deploy qMR Lab in the cloud. And we're able to use high-performance computing to actually make the analysis fast. This is just a very brief video to show you how qMR Lab works. I'm going to scroll through it. I'm not going to let you listen to it, but I just want to show you what the graphical user interface looks like. So here are all the different methods that we currently cover. And as I showed you, these are the functionalities. This is what the MATLAB interface is like. You can choose your module. You can load the data. You can explore the data, visualize it. And then you can perform data fitting analysis to see how your fits worked. And you can do different kinds of visualizations, histograms, sensitivity analysis, and single voxel simulations. So this is something that is still in MATLAB, but as you saw, a lot of the community is actually still using MATLAB. 80% of the papers in MRM are in MATLAB. So we thought this is the community we first need to serve, and maybe there will be a Python version coming soon. Now, open source, many people associated with not-for-profit, making work freely available, which is a noble goal, and it's great. But the work is not going to be properly used unless there is a commercial tie-in. So there is a company called HeartVista, which is founded by Juan and Bill. They were my colleagues at Stanford, graduated a couple of years before me. On the board is also Bob Hu, a cardiologist, and my PhD mentor, John. And this company is actually really interested in incorporating qMR Lab into their workflows. What does that mean? Well, here it is. This is a Siemens scanner. It's a 3T Siemens Skyra at the Montreal Heart Institute, where I do my research. And then here is a platform developed by HeartVista, by the Stanford startup. And RT-Hawk basically bypasses all of the proprietary stuff from Siemens, the pulse sequence, the control of the gradients, and does all of the acquisitions using its own open source pulse sequence development environment called SpinBench. Using HeartVista, we're actually able to deploy qMR Lab on any Siemens or GE scanner that has the RT-Hawk platform. So what does that look like? Well, here it is. You can do some real-time acquisitions. And here it is. You can change the field of view. You can change the angle at which we're doing the acquisition. Here you go. You can actually take a phantom and you can scan it. This is the NIST phantom that I told you about. It's distributed at many different sites around the world. And this is what the image looks like when it is actually acquired through the RT-Hawk platform. This is a comparison, the acquisition from Siemens and the acquisition using a sequence that we published publicly on GitHub. If you want to stare at it, Siemens is still superior. Their contrast is better. But this is really a first draft. And this particular acquisition was done only in about 24 hours after we got the RT-Hawk system at the Montreal Heart Institute. Afterwards, the pandemic happened. The institute closed. My student Aga went back to Turkey. And finally, now he's back in Montreal continuing this work. So a lot of this you're going to see, it's very preliminary results, but it's very, very promising. So here are the results. here is that NIST phantom acquired on a Siemens system and on the RT-Hawk system. And you will see that for very low T1 values, we have an excellent match. For very high T1 values, RT-Hawk underestimates the true values, and Siemens is better at obtaining those. However, we think this is a very simple spoiling problem. We're actually fixing it. We were now able to overestimate the values. So I think there's going to be a version 2.0 coming very soon. And we can run the sequence both on Siemens and on GE. This is the point where I would like to give credit to another group that have a product called Gamma Star. Here it is. It's really exciting. We recently had an interview with them for the MRM Highlights Initiative, and they're actually able to deploy this vendor-neutral sequence on 3 different vendors, on Philips, GE, and Siemens. The approaches are similar. One of the differences is that they actually take the proprietary language and they try to interpret it using the Gamma Star approach, whereas we write our sequences in SpinBench, which is an open source pulse sequence development environment by RT-Hawk. And then we don't need to do any kind of compiling in IDEA or EPIC. It's RT-Hawk that actually does that work. So here it is. This is what it looks like. We have all of the control of the sequences right here using the SpinBench system. You can actually fork this particular sequence on GitHub. Here's the link. And if you just click on this link, it will actually take you to a notebook where a lot of these visuals are available and people can actually interact and change the parameters. Importantly, we actually use the BIDS brain imaging data structure format. And this is what enables us to standardize a lot of the acquisitions across sites and across platforms. It's a single call, so you can actually clone this repository. And this is what you're going to get. This is the RT-Hawk control. You can control the parameters such as field of view, flip angle, TR, TE. You can create a pulse sequence schematic. You can see what the shimming does. You can see all of the pre-scan routines. And here is the actual variable flip angle T1 mapping routine. Then what we do is we containerize a lot of the post-processing using something called qMR Flow. It's a companion piece to qMR Lab. We use the BID standard, and here it is, a T1 map is eventually generated. As a proof of concept, I think this is really exciting because the idea is that you have a Siemens platform and a GE platform. We know that there's differences between them. So here's the black box from GE, here's the black box from Siemens, and here is our vendor neutral platform that just kind of bypasses all those black boxes, feeds the data into RT-Hawk, reconstructs, and uses qMR Lab to give you 2 identical brain maps, quantitative MR maps on both systems. To make this be feasible, it's not enough just to deploy a pulse sequence on the scanner. That's part of the work. And here it is. The PulseSeq folks, the Gamma Star folks, RT-Hawk have tackled this issue of vendor neutral acquisitions. But then you take the data, what do you do with it? You need to reconstruct it. There's a lot of different tools that can reconstruct raw MRI data. Once we have the reconstruction, then we have to pre-process. There's lots of different tools that can do the pre-processing. Once we have the pre-processing, the data goes into analysis. And of course, there's lots of different tools, many of these open source, for analyzing MRI data. So how would a workflow like this work if we were using qMR Lab? Well, there's a couple of data standards we have to worry about. The ISMRM raw data standard, that's very important, and the BID standard. There it is. We have a workflow engine that can take data from the scanner. It will wait for the ISMRM raw data format to be converted properly. It will reconstruct the data into a standardized BIDS format. It will pre-process it using elastics. And then finally, the processing will happen with qMR Lab. All of this is orchestrated by industry-grade workflow engine called Nextflow, where you take the data, you process it, and all of the steps are properly containerized to ensure that the environments in which the processing is happening is vendor neutral and system agnostic. So I realize that this is a lot to take, but this is not the work that any of standard MRI researchers need to worry about. This is a technical issue that needs to be overcome. And what we're trying to make is a workflow that's really easy for other people to use, even if they don't know what a container is, even if they don't know what the ISMRM raw data standard is or the bit standard. All they need to recognize is that without this, all of these different pillars are a source of variability. And if you want to get your numbers, well, it's just not going to be reproducible. So we have a recipe. We think people are going to use it. And I draw a lot of inspiration from this book. It's called The Cathedral and the Bazaar. It's a book about how computer systems, software in particular, used to be built and how they're currently built. It's the story of Linux and how basically we used to work in this cathedral, building things on top of each other. But then at some point, the work got distributed and we created a bazaar. And there's Linus's law, which is a really nice summary of what I've been trying to do, which is given enough eyeballs, all bugs are shallow. So it makes a lot of sense to make it distributed, to make different parts of the system work together, but also to make the black boxes visible to researchers. So where are we going? I hope we're going towards the first MR bazaar. There's qMR Lab, there's HeartVista. We're working with Siemens. And what we would like to do is treat the MR scanner the way the cell phone has evolved. You have a system, you have the hardware. It used to be all about the performances of the hardware. Eventually, it became about the App Store and the apps that were running on it. And now you can get the same app running on Android or an iPhone. I would like to get that same kind of functionality on Siemens or GE or Philips or Canon. And I think it's a worthwhile goal. We're working towards it, but we need industry partners and we need also buy-in from colleagues that are performing this research. Also, we need to think about standards. It's really easy to tell a lofty story, but unless it's backed up by something that a lot of people, a big community has agreed on, it's not going to work. And that's why this whole aspect of community building is really, really important. So I have about 10 more minutes to tell you about all of the community building efforts that we're making to make this vision reality. One, I want to tell you why we're focusing on T1 mapping. Well, I think that unless we fix the field of T1 mapping, the rest of quantitative MRI doesn't stand a chance. Many other quantitative MRI routines are actually built on top of T1 mapping. And also T1 is such a fundamental property of MRI that unless we can measure that, I don't think it's worthwhile to measure other things. There's another aspect of this, which is, well, T1 mapping is useful for many things. For example, some people will say T1 mapping is myelin imaging. That's a big debate. I've reviewed and responded to reviewers about this for a long, long time. And then now I actually have something to show that can convince people that yes, T1 mapping is a good substitute for other myelin imaging techniques. This is an article that just came out in eLife. That was like 2 weeks ago.
Nikola, this is Jeff. I'm a postdoc working with Ellen, have done some quantitative MR and have a lot of respect for what you're working on. This is really impressive. I wonder if you have any words about regulation. It seems like part of the reason or part of the way that the vendors kind of defend their silos, right, is a regulatory comment, like how would the FDA approve open source? I'm also thinking about all the questions that come up with regard to driverless cars and liability, and that these are really hard problems on how you take open source tools and then have them make, or have them involved in very consequential decisions. Just wondering if you have any thoughts.
Yeah,
maybe can
absolutely.
I add on to that, to the whole billing issue on the clinical side, because that kind of adds on to or fits in with Jeff's comment.
So one, absolutely. We've thought about this a lot. So for now, we all know, in our research, this is okay. If we're going to take it to the clinic, well, we have a problem. Two things. One, HeartVista, the RT-Hawk system from HeartVista, is FDA approved. Not for all of the applications, but for some of them. And every time you add a new feature, it will go through the same process, but there is a way to get RT-Hawk in a clinic right now, and it will be able to do on one-click cardiac MRI acquisition. Now, the question is, how do we go about actually getting an open source pulse sequence on RT-Hawk and getting it FDA approved? And this is where version control comes in. Open source is no worse than anything else as long as you're able to properly freeze it. Once you freeze it, once you say, this is the deliverable, this is what I'll get the FDA approval on, you can containerize it, and you can put it on the system just like any other application. So while the development cannot continue because of the FDA approval, at some point you need to say, okay, this is a version that's FDA approved. I don't think there is any problem of taking a sequence that used to be public and just freezing it and putting it on the system. Now, vendors will tell you, but yeah, that compromises IP. But I think there is a way forward that doesn't really care about the vendor story. It's really more about, can you trust this sequence? Yes, you can. It's frozen in time, and it was developed publicly. And at the moment, it's deployed on every RT-Hawk system around the world.
Thank you so much. That's really helpful.
Yeah. And my computer is back, so I'm happy to take another question or two if, seems like this is a good stopping point, but I'll join from the other computer and see if I can wrap up.
So maybe Nikola, as you're thinking or starting that, I'm curious, Boris probably mentioned too, there's, these multi-site studies that are ramping up to do imaging across the lifespan, many of which are wanting to do quantitative MRI on multiple platforms. Where do you think the status of that really is, or where products like qMRI fit in that spectrum?
Yeah, so qMRI, it's synthetic MRI, is that correct?
Yeah.
So I think there's a very nice follow-up right now in the presentation where I think it will hopefully help you position synthetic MRI. Not to keep you in suspense, I think the fact that synthetic MRI is properly standardized across vendors is an excellent development. And I think they can make a strong case, as you'll see in the meta-analysis. So just a second, I need to start the slides. But they can make a perfect argument that, look, all of these different methods are comparable, and ours is properly standardized. Now, it's not very open, and I think that's one of the disadvantages. And the other one is there haven't really been proper histological validations of the technique. But in my opinion, the fact that it's standardized goes, really well with what I'm preaching. Even though synthetic MRI might not be as transparent as I would like it to be. Here I am now on my computer. Even though a synthetic MRI is not as transparent as I would like it to be, it's out of the box, it works, and as long as they provide proper version control, I think it's a reasonable approach. And here it is, what I would like to show you regarding the approach for myelin imaging. We created an interactive meta-analysis that shows all of the different papers that have done histological validation of myelin imaging. This is the link right here. And let me just guide you a little bit through it, just so you see what it looks like. Now that the computer shut down, I actually can't show you, I know that. But here's the non-interactive version. We take a look at different methods, diffusivity or diffusion, magnetization transfer, and relaxometry-based approaches. And you will see that the R-squared values that you expect to get as correlations between myelin and an MRI metric of myelin, it's kind of low. It's less than 0.5 most of the time. And I think a conclusion we can draw is that most of the myelin measures for MRI are statistically interchangeable in relation to the underlying histology. The literature does not suggest a universal MRI-based measure of myelin. And informing future studies is difficult due to large prediction intervals. Now, we actually have the statistics to show that. And we were working with Tom Nichols, who is a leading statistician in the UK. And this is my favorite map that I will use now for every review I will ever do or when I get reviewed. We just tried it, it actually worked. Because this graph shows you which technique can be exchanged for which other technique. So for example, here's MTR right here, and here's T1. You take a look at the cross-section between MTR and T1. Turns out that they're not statistically different, at least in the meta-analysis sense. And this is a perfect proof that MTR and T1 are just as good in measuring myelin. They might be measuring different aspects of myelin. Maybe MTR measures lipids and T1 measures water. But in the end, because the correlations are kind of moderate, not very high, they're pretty comparable. So next time somebody says, why did you use T1 for mapping myelin? I can say, well, unless there is a truly biophysical reason why you think one would be superior to the other, statistically, they're not different. On the other hand, if you have diffusivity, well, diffusion measures are not as good. And you will see that, wherever you have these violets, that means MD cannot be a replacement for magnetization transfer or for myelin water fraction imaging. So the link is there. I was really hoping to guide you through this, but we had some technical difficulties. I do encourage you to take a look at that because I think it's a really, really important contribution. Finally, I want to tell you a word or two about the Canadian Open Neuroscience Platform. This is the platform that's allowed me to do a lot of this work. It's a big collaboration between 33 institutions. As I said, I'm in charge of its communications platform. We recently published a blog post, and it's about what we hope to do going beyond Plan S. Plan S, the initiative in Europe that mandates everybody publish open access. We think that that's a worthy goal, but it's not enough because as much smarter people than me have said in the past, David Donahoe in particular, a published research article is just advertising for the work. The actual work is what lies below the surface. The data, the code, the reviews, the simulations, the hypothesis, the raw data sets, and these are the kinds of things that we want to expose. So we have published a recipe for a reproducible analysis, and we would argue that your code needs to be on GitHub, the data needs to be publicly available, the environment needs to be Dockerized, and you should push more than just a static PDF in the form of interactive plots and analysis. How would that look like? Well, this is our project. It's called NeuroLibre. It actually has its soft unveiling tomorrow. And this is going to be our vision for what a publication of the future would look like. So here it is, a PDF compatible document that we're all used to seeing. And that's the first layer of what we hope to publish. Here's the second layer, figures that are dynamic, so you can actually observe the data points. A third layer, you can explore the phenomenon. There it is. You can explore the signal as a function of inversion time, or you can interact with the real world data, such as these brain acquisitions using inversion recovery. You can see the code that generates their outputs. So here it is. Everything that's been done to get these brain images is right there in the book. And finally, you can run the code that generates the outputs and you can execute it. Now, this fifth layer, the reproducibility layer, is what we're really trying to push with publishers. The eLife story is an example. We also have partnership with Plosh Computational Biology, and we're working with ISMRM. ISMRM recently gave us an award for this. It was the Africa Challenge of the Junior Fellows Symposium. And the title is All You Need is a Browser, Eliminating Barriers to MRI Education with Open Source Interactive Tutorials. This was won by Mathieu, by the software developer in my lab. And this is our vision for how I would like MRI research to work. Rather than jumping through hurdles, data acquisition, data pre-processing, analysis, finally publishing, I'd like it to be kind of a more leisurely stroll through the park, where you go through industry, you partner up, you use open source tools, and eventually publish it using innovative publishing platforms. To do that, we're trying to get a community behind all of these initiatives. There's a podcast with the Canadian Open Neuroscience Platform where we interview a lot of open science advocates. MRM Highlights, this is an initiative that I started 5 years ago, and I recently stepped down. And I think that was the best thing that happened to the initiative because my software developer picked it up. And now the emphasis on reproducibility. We actually highlight papers that exhibit good reproducible research practices. We recently organized a symposium, a webinar, which was a collaboration between the Canadian Open Neuroscience Platform and MRM Highlights. Here's the group photo of the people that turned on their camera. There were more. We had about 100 people attending. But you will see Peter Jezert, who is the current editor-in-chief of MRM, Matt Bernstein, who was the past editor-in-chief. You'll see a lot of familiar faces. Peter Bandettini, former editor-in-chief of NeuroImage, currently working on the OHBM publishing platform called Aperture. Borjan, right there, he actually joined us for this. It was very good to see him. And this was a very productive discussion. We're about to publish this on the MRM Highlights channel. We organize a hackathon. So there it is. That's my shirt. This is something that happened in Montreal in 2019. We were planning to have another one in New Zealand right before the ISMRM meeting got canceled. But I think we do need this contribution. MR professionals working openly to develop new techniques, new protocols, new sequences. And then we also have these meetings that happen weekly. We call them Open Science Beer or Open Science Beverages to be a little bit more inclusive. Here's a cake we made for the 1-year celebration. This is the celebration at the INCF Neuroinformatics Conference that happened in Montreal in 2018. This is the Open Science celebration at the ISMRM. Many of you were probably there. This happened at the Phi Center in Montreal right after the conference. We had about 1,000 people there. And I think it was a really nice way to celebrate the community we're building. And here I am. I'm just going to wrap up with conclusions, which is that DMR Cathedral is slowly turning into a bazaar. We need to think outside the quantitative MR black boxes. Vendors have locked us up into systems that are not particularly reproducible. Quantitative MR analysis tools are revolutionizing research. And to ensure reproducibility, we need to talk about version control, standards, and licenses. That being said, community building is essential. As you've probably heard in the past, 20% of this work is technical and 80% is social. So I hope that we actually get to a point where we're all working together towards a common goal of transparent, reproducible quantitative MRI and hopefully MRI in general. This is my lab. Julien Cohen-Haddad is the co-PI. The lab is called Neuropoly. We're really grateful to the collaborators that are in the left column. And finally, all of our funding agencies. And the name of the lab is Neuropoly. You see this visual, you'll hopefully remember it. This is my email. Please get in touch. This is my Twitter handle. And we are hiring. So if anybody would like to continue their work with us, we would be very, very happy to talk to you.